Hi, everyone. Welcome to our webinar today, AI, Digital Twins, and the Future of Clinical Research. This is part one of a three-part series sponsored by Unlearn AI. I'm Arsalan Arif, the publisher of Endpoints News, and it's my pleasure to introduce our presenter today, Charles Fisher, the founder and CEO of Unlearn AI. Charles has an approximately 30-minute presentation for us today, and then I'm going to pose questions from the audience to Charles for the second half of the webinar. So please hit that Q&A button at the bottom of your screen down there as soon as a question pops up in your head. I'm going to work to get as many of them to Charles in this session as possible. And now, without further ado, I give you Charles Fisher.
Thank you very much, Arsalan. I'm really looking forward to the discussion at the end of this presentation. So today, I'm going to talk about a really, I think, exciting technology that we've been developing at Unlearn. And what I'm really going to focus on is how biopharma companies can run clinical trials in half the time using half as many patients. And in order to understand this problem, we first need to kind of go back and ask, what is it that a clinical trial really does? So every clinical trial, or most clinical trials, aim to determine if a new treatment performs better than a standard of care. And to do this, they compare two potential outcomes. So on the one hand, we say, what would happen to this particular patient if they were to receive a new experimental therapy? And then we say, on the other hand, what would happen to this particular patient if they did not? And then what we want to know is how different would that patient's outcome have been if they had received the placebo instead of the new treatment? What is that difference? And there's something that we call the fundamental problem of causal inference. And this fundamental problem is that this quantity, this difference between potential outcomes, is not something we can observe. We can give the patient the drug, or we cannot give them the drug. We cannot do both at the same time. So that makes understanding treatment effects difficult. But maybe there's a way that we can use computational methods to try to answer this question in a more direct way than we typically do. And it turns out that engineers encounter this same problem all the time. And they often solve it using a kind of technology called the digital twin. And so here I'm drawing on an example from this article by the BBC about how GE uses digital twins in their manufacturing and monitoring of jet engines. The concept is relatively simple. We have a real jet engine, which has a number of different sensors on it. And we're going to take data from those sensors and put them into a computer model, a computer simulation that has been built using the blueprints of that engine. And then we can use this computer simulation to simulate how that particular engine would perform under normal conditions. And then by comparing what you actually see happen in the real engine that's out there functioning in a jet flying around in the sky to what you're simulating, you can get some understanding of how different operational conditions change the operations of that engine. So let me just dig into this analogy just a little bit deeper. Let's imagine that we have taken our blueprint and we've built a computer model of an engine that's on a jet. And then we can take data from that engine and build a digital twin that has all of the same characteristics of that engine. So here we have the actual engine operating at low RPMs and the digital twin tracking it, simulating how it performs at these low RPMs. And now we're going to make some kind of intervention. So we're going to step on the gas and we're going to rev the engine up to higher RPMs. But before we do that, we can take our computer model and we can simulate using the digital twin what would happen if instead of hitting the gas, we were to continue operating that engine at low RPMs, and we can get this prediction. And then we can look at the real jet engine after we've stepped on the gas, and we can observe what happens to the temperature of that engine over time. And we observe this difference, that the real engine operating at high RPMs, the temperature increases much more rapidly than in the digital twin. And so the difference between what we observe when we perform this intervention on the actual engine and what we predicted from the digital twin tells us about the effect of hitting the gas on how this engine operates. And that concept is actually very similar to what we do in drug development. So is there a way that we could take this idea of a digital twin that engineers use and bring it into drug development and try to build digital twins of people? So unfortunately, an engineer has a blueprint for an engine because they designed it and they built it. And we don't have blueprints of human biology. In fact, human biology is extremely complex. And even if we wanted to try to build some sort of computer model to capture all of the intricacies, it'd be basically impossible. So just to bring that point home, this is a protein-protein interaction map of just 33 genes that are associated with Alzheimer's disease. And it has hundreds of different interactions between all these genes. So try to imagine scaling this plot up, this graph, to the 20,000 or so genes that people have, and think about how could you possibly build a model with that level of complexity. Something very daunting, and that just seems out of the realm of at least today's technology. So we can't build digital twins of people from the ground up. It's just something that's out of the realm of possibility today. But maybe we can build digital twins top down. So there have been tremendous advances in artificial intelligence over the past 10 or 15 years. And these are actually real things that people are using every day. You may not even realize it. Your cell phone translates languages. There are self-driving cars. I live in San Francisco. There's self-driving cars all over the streets. And so there are real advances in artificial intelligence that may make this possible. And one of those particular kind of advances is in something that we call a generative model. And generative models give us the ability to create digital twins without actually needing to go bottom up, without needing the blueprint. And I want to present one more final analogy before we actually get into work on clinical data. And this is an example of how a generative model works, drawing from a research study conducted by NVIDIA. And so in this case, they took a large database of images of just people's faces. And they probably have millions or maybe even billions of these images of people's faces. So a big database of images. And then they trained a kind of artificial intelligence model, something in this case called a generative adversarial network. And this model learns to draw new pictures that are photorealistic images of imagined faces. So these pictures on the left are real people, and these pictures on the right are not. They are drawn entirely by a kind of neural network that's learned how to represent pictures of people's faces and then how to draw new faces that have those same characteristics. And in fact, after the webinar, if people want to play around with this, you can go to a website called thispersondoesnotexist.com, and you can actually generate your own faces using this machine learning model. And so what we do with clinical data is actually very analogous. We build a generative model that's trained on longitudinal patient information. So we can collect a whole big database of clinical records from actual people, from actual patients. And that could be from historical clinical trials. It could be from registries, or even in certain cases, if you can get high enough quality data, directly out of the EHR. And so we can create this database and use it to train a kind of generative AI model that will now create new clinical records, which we call digital subjects, that are not actual clinical records of any real person on earth, but which capture all of the characteristics of clinical records of real people. So these would be like completely anonymized, simulated clinical records of patients. And then we can use this type of generative model to create digital twins. So let's walk through that process in a little bit more detail. And this whole 30-minute presentation today is going to stay fairly high level, and subsequent webinars 2 and 3 will go into a bit more detail about certain things. So how do we use artificial intelligence, generative models, to create digital twins for patients in clinical trials? So step 1, and everyone who actually has ever had the pleasure of trying to work with lots and lots of data and do data science knows that this is actually a very, very difficult step, probably the most difficult step in the process, is an individual clinical trial is relatively small. You might have maybe a couple of hundred patients, say from a phase 2 trial. And that's probably too small to train a kind of big generative model on. So we need to take data from many, many different clinical trials and registries and EHRs and integrate it and then clean it up. So we take data from lots and lots of clinical trials and registries. We extract the information from patients in those trials who received placebo or standard of care. And then we spend an incredible amount of time and effort cleaning those data, integrating them and harmonizing them so that we have a highly curated data set that's suitable for training a machine learning model. And then we're going to create a type of generative model that's able to generate digital twins by taking in the baseline data, so the data collected from a real patient at the start of a study before they receive a treatment, and that can take those data and then simulate how every single one of those different measurements may change over time to create a type of potential clinical trajectory for what would happen to that patient if they were to receive a placebo. And we're going to call this a digital twin. And how do we actually do the training? So here we have a model, may have a few thousand different parameters in it that we need to tweak those parameters until it's able to generate these potential outcomes, these digital twins that look realistic. And the way that we're going to judge whether or not these things are really being realistically generated is that we're going to take a digital twin or an actual patient record, and we're going to pass it to some type of algorithm that's designed to tell those two things apart. So we have something that's generating digital twins, and we have another thing that's critiquing the digital twins that are being generated and asking whether or not they are similar or different to real patients who are receiving placebos in these clinical trials. And if we're doing a really, really good job, then you shouldn't be able to distinguish a real patient who received the placebo in a previously run trial from a digital twin, which is a prediction of what would have happened if they received the placebo. Those two things should be essentially identical. And so when we use this classifier to tell them apart, we don't want to be able to do so better than you can predict the outcome of flipping a coin. So we should be able to do this no better than 50%. It should just be randomly guessing as to which is which. And why is that helpful? Well, if a digital twin is an outcome, then it is equivalent to a placebo subject in a real trial in some quantitative way, then we can use them as information to inform a understanding of control arm behavior. And I'll describe that in a bit more detail in a few slides. But first, how does this actually work? What does the process look like in an ongoing clinical trial? So before the clinical trial actually starts, we will have collected and curated all of these data. We will have trained this generative model. And that's all set aside before the trial starts. And then when you're actually working in a clinical trial, some particular patient enrolls. And on their first visit, a large number of different measurements are made characterizing what that patient looks like before they receive their first treatment. And these baseline data are entered into this generative model, which then creates a digital twin. So it predicts how all of these different patient characteristics may change over time. Then the real patient is given a particular treatment, and they go through the trial and data are collected. And at the end of the trial, you have an observed patient record for that real patient. So now we have basically access to these two potential outcomes. One of them is observed and one of them is simulated. And so we can estimate a treatment effect simply by comparing these two potential outcomes. So we can take the clinical record that was observed from a real patient who has received the treatment and been followed up over the course of the trial. And then we can subtract what we think what the simulation predicts would have happened to that patient if they had received the placebo. And the difference between those two potential outcomes tells us the effect that that treatment had on this particular patient. Now, of course, this is a very granular, detailed estimate of the treatment effect, but we can aggregate up all of these different components to understand the primary or secondary endpoints of a trial. So we've initially focused our efforts on neurodegenerative diseases. This approach, however, really, because it's entirely data-driven, could be applied across different indications. It's entirely driven by the data that you put in. But our initial flagship product that I'm going to go into a bit more detail about creates digital twins for patients with Alzheimer's disease. So why Alzheimer's disease? Well, I think that there are a couple of important reasons. So first, Alzheimer's disease is an area of tremendous unmet need. Patients are really waiting for a disease-modifying therapy that can make them either slow the progression of cognitive decline or reverse cognitive decline. But we've had tremendous difficulties developing drugs and new treatments for Alzheimer's disease. Not only in the fact that there's been very, very high failure rates, but these clinical trials are very expensive and long to run. A typical phase 3 trial in Alzheimer's has more than 800 patients, takes more than 4 years to run, and costs something like $200 million. And that really slows the progress that we can make towards defeating this disease. So is there some way that we can leverage these types of new machine learning algorithms to speed up this process? So the first part of our process for training this type of generative model is getting data. So let's talk about the data that we actually have for Alzheimer's disease. You know, over the last 15 years, there have not been really any successful clinical trials, or there have not been any yet approved therapies. It is arguable whether there have been some positive therapies. We'll find out more about that soon. But that means that there's a wealth of information, a large number of these clinical trials that we can use to inform future clinical trials. So we've, working with the critical path for Alzheimer's disease, we have created a data set that has approximately 7,000 patients with Alzheimer's disease, totaling 36,000 patient visits, taken from 21 different studies. And the majority of those studies were placebo-controlled, randomized controlled trials. And we are only taking the patients from those studies who received the placebo. We are also incorporating some additional data from observational studies, but that makes up a small component of our overall data set. About 25% of these patients had mild cognitive impairment, 75% of them had AD, and this is covering many key variables, things like ADAS-Cog and MMSE and CDR sum of boxes, the scores that people use for measuring cognitive decline in these types of trials, as well as labs and vital signs, demographic information, and biomarkers. Then we have to go through the process of actually training and validating the model. And so the first step in this is, now that we've taken this data set of historical trial data and registry data, curated it, we're going to divide it in two. And we'll take 80% or so of our data, and we will use it to train this generative model. And we will leave out 20% just to test and say, how are we doing at predicting or creating these digital twins for patient data that we have not seen before. And this type of cross-validation is sort of the first step in validating a model that people in machine learning research typically do. And so once we go through this process, we then have a trained generative model. And I'm not going into a lot of the details of our machine learning methods. For those that are interested, we have a number of peer-reviewed publications on our website that describe the particular type of machine learning neural network that we use for this type of problem. But I'm not going to go into the technical details of that today. Although if people are interested, you can submit a question and maybe Arsalan will ask it at the end. Okay, so we get through this whole process. We've now kind of defined what a digital twin is in the abstract. It's a clinical record. We have all this patient data for Alzheimer's disease, but maybe you're wondering, what would a digital twin really look like for a patient with Alzheimer's? So let me show you. This is an example of a clinical record that we have generated using our AI model trained on this historical data set of patient data for Alzheimer's disease. So we have a whole bunch of different variables at the top, things that are not necessarily changing over the course of the trial. They are fixed based upon how that patient looked at their first visit. Then we can measure all of these different components of the cognitive scores, all of these lab tests and clinical things that are used for safety. And in a digital twin, the data at time 0 would be determined by the real patient. They would be exactly the same values that we measured on that particular patient. And then we are simulating how each one of those different characteristics will change in 3-month intervals over the course of a clinical trial. So we are talking about a detailed medical record with the same level of detail that you get out of real patients in clinical trials, not just a coarse-grained simulation of a patient, but a really detailed clinical record that we can actually export directly into CDISC formats. Okay, so that's the level of detail we're talking about, but how accurate are these models? Can we distinguish them from real patient data? So I said before that the way we're going to train these models is to have a classifier and ask, give it a real clinical record and give it a digital twin and ask it which is which. And so we're going to do that, but we're going to specifically do it looking at the patients we left out of the training process. So that smaller data set we're using for validation. And if we do that, we take this model and we give it actual patient and we give it a digital twin and we ask it to tell the difference between those things. It can tell that it is correct in telling which is which 51% of the time. So that's essentially the same as a coin flip. So even though we have these incredibly detailed, rich, longitudinal generated patient records, using all of that information, it's still not possible, even in that case, to really tell these digital twins apart from actual patient records. And that's at that level of individual detail. But maybe we want to roll this up and ask a more coarse-grained question about something like a primary endpoint. So here's an example of actually now a completely different, very out of sample, looking at other studies, test of how well these digital twins can predict control arm behavior. So here along the x-axis, we have the time in the clinical trial. And along the y-axis, we're plotting the change in the CDR sum of boxes scored during that clinical trial. And we have two different studies. One on the top, which was conducted in patients with mild to moderate Alzheimer's disease, the triangles. And another on the bottom, the circles, conducted in patients with mild cognitive impairment. And we are plotting in black the actual behavior of the control arms in those studies. And in blue, we are plotting the predicted behavior of those control arms obtained from digital twins. So these digital twins are computed, again, at that very high level of granularity, that high level of detail. We're just showing one particular measurement in this plot. But they are computed entirely from the baseline information. So from this baseline information, we can predict how these different control arms in these trials will progress very accurately into a high degree of detail. So digital twins for Alzheimer's disease are very detailed and very accurate. But how can they actually be used to improve clinical trials? So first, I want to introduce just a bit of terminology. So a digital twin acts like a control arm for a single subject because it has the exact same baseline measurements as that particular subject. But in clinical trials, we often want to ask questions about groups of people. And so we need to refer to a group of digital twins. And so we're going to refer to the cohort of all of the digital twins that have been generated for all of the patients in a clinical trial as an intelligent control arm because it's created by an artificial intelligence algorithm. And what I'm going to argue, and we're going to present a lot more data on this in the next webinar, is that incorporating these intelligent control arms into clinical trials can lead to much more robust, efficient, and personalized drug development. So we can increase the statistical power of the trial, generate more confidence in the results, decrease the number of subjects, and thereby accelerate clinical trial timelines. And because each digital twin is perfectly matched to a single individual patient, we can start to drill down and understand how individual patients in a trial are responding to a treatment. So let's think about one potential use case. Let's imagine that you're running an Alzheimer's program and you want to run an early stage proof of concept study. Or maybe you've finishing up a randomized controlled trial and you're going to have an open label extension period. And in both of those types of studies, maybe you're running a single arm proof of concept or an open label extension, every single patient is receiving the active treatment. And if every single patient is receiving the active treatment, then what do you compare to? Well, you could compare to some historical population or historical average, but that's not necessarily the same as the particular patients in your trial. So one thing that we can do is we can create an intelligent control arm by making digital twins of every single one of these patients, and then use the intelligent control arm as an external comparator. And running trials in this fashion enable us to essentially half the number of patients that we need for a clinical trial. So some of you may be very familiar with the idea of external comparators. And even though we have a sophisticated generative machine learning model that generates these very, very detailed clinical records, there is still a potential issue. So let's imagine we're running a clinical trial where we have an external control. So we can imagine that every patient in our study is receiving the treatment, and every patient not in our study, in our external control, is not. And we believe that this treatment may cause a change in the outcome. So it may change the rate of progression for these patients with Alzheimer's disease. And we've measured all of these different characteristics. So all of these characteristics about the patients that we've measured, we can account for. We can ensure that they are perfectly balanced between the intelligent control arm and the external comparator and the treatment arm. But what about things we haven't measured? Well, things we haven't measured, we can't control for because we don't know. They are the unknown unknowns that have the potential to bias the trial outcome. If they are related to the change in outcome, and they also are different between the patients in the active arm and the patients in the external control. And there's actually only one way that we really know of that's able to account for these unknown unknowns and make a study more insensitive to it, and that's randomization. So randomization is a tool that we have for making inferences that we draw about treatment effects to be insensitive to unknown unknowns, to unmeasured patient characteristics. And before I showed a way that we could add an intelligent control arm to a single arm trial and use it as an external comparator, but we can also add an intelligent control arm into a randomized controlled trial. So the way that this works is that when a patient enrolls into the trial, a digital twin is created from their baseline data, which is collected before they receive a particular treatment. And then that patient either goes on to receive the active treatment or they go on to receive a placebo. Regardless, we've already created a digital twin. So there is this intelligent control arm that exists for every single patient in the trial, whether they are in the active arm or the control group. And what that means is that we now have an internal reference. We have a group of patients who actually received the control, and we have a group of digital twins, which are predictions for what would have happened if those patients did receive a control. And by having this internal reference, we can ensure that we can increase the statistical power and efficiency of a trial while remaining insensitive to potential confounding variables. And this is actually a very powerful idea because it's not a small change in how you can design a clinical trial in this way. So we can show that for Alzheimer's disease, adding digital twins onto a randomized controlled trial enables one to decrease the size of the control group by up to 75%, while retaining all of the characteristics of a randomized controlled trial that you want, while ensuring that you have good operating characteristics and that you are insensitive to confounders. We can get the best of both worlds, blending the robustness and the rigor of a randomized controlled trial with the efficiency and speed of a single-arm clinical trial by leveraging digital twins and intelligent control arms. And in the next webinar in this series, we will walk through a detailed example demonstrating by retrospective analysis of a completed clinical trial where you can compare the results of the just plain old RCT to the results of the RCT with an ICA for Alzheimer's disease, to really walk through how these methods work and to present clear evidence that demonstrates that these intelligent control arms can benefit multiple types of trial designs. And that more detailed second webinar in the series will be taking place on October 21st. So thank you very much. And now I'm really looking forward to this discussion with Arsalan.
Thank you, Charles. Thank you, Charles. Judging from the number of questions and comments that we received. I think you got the audience's attention. So let's get to the questions. And I actually encourage the audience to keep asking because if there are any questions that I'm not able to pose to Charles here in the next 26 minutes or so, we'll definitely work to try to get back to you by email or something. And so Charles, I got so many questions here that I've kind of bucketed them a bit here. And I'm going to try to get to all of them. There's just so many. There's so many keep coming in right now here. But here are the groups: intelligent control arms versus external control arms. There are a lot of questions on how the model or models themselves are made and how those models are fed. Having a lot of questions on placebos here. And there's some really good questions here, which on data privacy. So let's get to some of the intelligent control arms versus external control arms questions here. I'm going to read two questions to you because they're a little bit similar, but you might be able to tackle them both together. So let me read the first one here. What is the similarity or difference of this and using real world data as historical controls? And what biases would the AI models create? Okay, that's the question 1A. Question 1B is, how does this technology differ from other technologies on synthetic control arms? Some of these technologies have led to FDA approvals for indication expansions. So just those two questions right there for you, Charles.
Yes, they're super different. The answer is that they're super, super different, right? So, and they're different really in a couple of different ways, right? So the first is really the source data. So let's start off with the source data because you mentioned about real world evidence. And a lot of times when people talk about real world evidence, they're talking about data, pulling it out of just routine clinical care, and then using that to draw inferences about patients, or in this case, as an external comparator, right? And the thing is that we believe in data quality first. Data quality is the most important thing. And so we are starting with a foundation of data collected in clinical trials and using sort of registry and observational study data sort of as like the cherry on top. So what that means is we're starting with data that were collected for regulatory purposes in the first place. They are rich, they are structured, we have a lot of follow-up for all of these different patients, right? So that's one difference, is in the data sources that we're talking about. But then you actually get to the question of, well, what do those methods do? Well, what they do is they take an actual real person in your study and they say, well, let's find the most similar person that exists in our external data set. Now, the most similar person could actually be extremely different. They don't necessarily match exactly. They actually often match very poorly. And so people use a method. And let me give you an example. Let's imagine that I'm working on Alzheimer's disease. I start off, I say, well, I want a patient. My patient is a man. So now I go, half my data set is gone, right? So half my external is gone. And I go, well, I want to be 65 years old. It's like, oh, now it's a much smaller group. And then I go, well, I want their ADAS-Cog word finding score to be a particular thing. And then I want their MMSE. All of a sudden, there's no patients who have all those different characteristics. It does not exist because it's a high dimensional space. And so what people do is they reduce all of that complexity, all of the information about a patient down to one number. And that one number is called a propensity score, and they match on that. But when you do that reduction, you throw away a huge amount of information, and you end up with a patient who is similar, but not the same. And we use all of the data for every single prediction that we are making, because all of the data are informing the training of the machine learning model. So no data is ever, it's all in there and it's all being used for that particular patient. In addition, the generative models that we use are probabilistic. This is also a fundamental difference, right? So when we make a prediction and we say this patient's ADAS-Cog score is going to increase by 2.5 points over the next year, we don't just say it's 2.5 points. We can give the whole probability distribution of how that score will change. So we can give the mean and the standard deviation and other information if you wanted it. It's not just one number. Whereas when you match by the propensity score, you get one patient who's similar and you treat that as the thing. So there's a lot of differences there in terms of the technology. And one of the last things here, so in addition to the fact that we're starting with regulatory grade data, that we are getting perfect matches across all of the different baseline characteristics of the patient, that we are leveraging all of the data using probabilistic predictions is we can generalize to new situations. So an example of this is actually, although all of the data that we are using to train these models come from studies that are usually 18 months in length. We actually presented this last year, I think last year, at the Clinical Trials for Alzheimer's Disease Conference. We can make predictions, simulate control arms for 5 years, and they're actually just as good as our predictions in sample. So we can generalize to new situations.
All right. Well, there's a follow-up question here. I think it might be similar here. I am confused why data from past trials is used here instead of EMR data. For a placebo, EMR data makes more sense to leverage than trial data, right?
So there are advantages and disadvantages to trial data and EMR data, right? There are major advantages to trial data. If anyone's ever worked with real trial data and then worked with EMR data, you'll see the quality difference is like infinite. It's like infinite quality difference. Yeah, I don't know that many people, and most people who go to the doctor every single 3 months and get a giant battery of tests performed. That doesn't happen. So this idea of having rich longitudinal information about patients from the EMR, that's not what EMRs are created for, right? So they just kind of doesn't exist. Now,
Okay.
the disadvantage of trial data is that it's very homogeneous, right? So the advantage of EMR data is now you can start to get more diverse patient populations. So when we are thinking about this, in constructing a data set to train a model, we're trying to put in the right characteristics. So we start with a foundation of that high quality data from historical trials. And then we add in a bit of EMR style data to get some additional diversity into the patient pool.
Okay. All right, still a bunch of questions here. So I'm going to move on to the next bucket over here. And this is just on the collection and preparation of the data, how you feed and build your model. How about this question here? How about the use of images? For example, CTs, MRIs for oncology or CNS trials. Would it make sense to include information derived from images in the digital twin approach?
Yes, absolutely. We do actually do that. So if when people, and this goes up online, people can zoom in really close on that slide with the clinical record in it, and you will see there are, for our Alzheimer's disease trials, some PET imaging derived measures. There's a couple of different ways that we could do that. What we're doing in Alzheimer's is including standard measures that are derived from images. You could also imagine using machine learning techniques directly on the images. That's something that, again, one could imagine, but that we haven't actually explored yet.
Okay. All right. I think you've already touched on this question here, but let me just ask you, why use a generative model to make a digital twin instead of the raw, highly curated patient data to act as the digital twin?
Yeah, again, because you can't match, right? You can't match on high dimensional characteristics. So we want to make these personalized predictions. We also want to get those predictions with the error bars on them, not just the number, but we want the error bars. And the generative model lets us get both of those things.
Okay. Can this technology be used to generate control arms based on a specific standard of care versus placebo?
Yes, absolutely. So in Alzheimer's disease, we have placebo. We also do have treatments like acetylcholinesterase inhibitors and the things that do exist. In other indications, that gets to be, where there are disease-modifying therapies available. That gets to be a bit more interesting than in Alzheimer's. But the only thing that we need is there needs to be data available for the treatment. If there's no data available for the treatment, then it's nothing a machine learning company can do about it.
Right. And so here's another question along those lines. And here's a comment and a question. Unless we want to use some poor scales, for example, MMSE, ADAS, COG, CDR, SB, how can we simulate digital twins with better measures like RBANS if we do not have any data a priori?
So our perspective in terms of these things, so you do need to have data on what you want to simulate. In the end, machine learning learns from data. If you have no data, you cannot make a prediction. Like that's just the general rule that could go far beyond this particular presentation.
Okay.
Now, we don't necessarily need an enormous amount of data for a handful of measurements. So let's imagine we have a model that already exists, and we just want to add in new measurements as they are becoming available. That can be done with a relatively smaller data set than you use to train the initial model. So these things can keep being updated and learning and getting better and adding new metrics as those things do become available, but they do have to learn it from data.
Okay. Let me ask another question here. Can you comment on the challenges with securing longitudinal patient data before they start on a treatment or trial, as this time series data is what is fed to train the model to generate twins?
Sure. So the model is trained with historical information, right? And it's longitudinal data. But it's not necessarily longitudinal patient from the person who we're generating the digital twin of, right? It's a longitudinal patient information from a cohort of people. And then for Alzheimer's disease, we only need one particular data point of that patient, which is the data that are collected when they enroll in the trial. So just the baseline data. That actually does depend a little bit on the disease. So people that are following our work in other disease areas may notice that sometimes you might need more information, depends on the disease. But for Alzheimer's, the baseline data are sufficient. And those are always collected in every clinical trial. So that's something that's relatively easy to do. If the question is about how do we negotiate with partners and things like that to get access to these other data sets, that's another interesting question, but maybe a little bit different from the actual technology or the science here.
Yeah, that's a business question right there. Okay. Here's a simple question. How are adverse effects monitored?
So there are two ways that so one here obviously is there's a real patient who is receiving a real treatment, right? So when you say, what are the adverse effects that this is happening on the real patient receiving the treatment, well, you observe them, right? So that's the first thing, is you observe them in real patients, right? In the digital twins, we have basically two things. One is that we are simulating the time evolution of all of the labs. So if you're saying you're looking at that granular detail about kidney function, liver function, all of those kinds of things, all of those things are there. And then in terms of adverse events, I mean, of course, this depends on how we do it for different indications. For Alzheimer's, we basically have categories of serious or not serious adverse events. And we do simulate the occurrence of those things in these digital twins for placebo patients. But again, the occurrence of serious adverse events, like if you think about something like ARIA, which is one of the adverse events that people are really concerned about in Alzheimer's disease trials, like the occurrence of that in a placebo arm is like basically it doesn't, right? Unless it's like maybe a false positive off of the read of the image, right? So it's a very, very low rate, but we do simulate those types of things.
Okay, all right. Let's go on to another bucket here, some questions on placebo. How do you account for the reality, okay, that the placebo effect and control group response can vary wildly between trials of the same or similar drug in similar populations?
Right. So I would actually dig into that question and separate out two different things. The first is that the control group behavior can vary widely between different populations or different studies, right? And so that's something that we actually see. Like our models, that's what happens, right? And for two reasons. One is that there is sensitivity to the baseline characteristics of the population that you enroll. If you enroll patients, I mean, I showed a slide there with MCI patients and with AD patients, and they don't have the same degree of progression, right? Those things are very different because the patient populations are different at baseline. However, it's also the case that our models are probabilistic and they simulate potential trajectories. If you run it over and over again and over and over again, you get a different outcome each time, and that gives you this distribution of potential outcomes. And we see variability in that distribution. So you actually can see this directly coming out of the simulations of the model. Second question about placebo. So one, we use a lot of placebo-controlled data for training the model. So that aspect of how patients potentially respond to placebos is something that's included. Because we also have some observational data, we can start to tease out how much of the control arm behavior is because they have received the placebo, and how much of it is just natural history or regression to the mean. And it turns out that in a lot of cases, natural history, regression to the mean, that's really what drives a lot of control arm behavior. And placebo effects are small, but that depends very strongly on what indication you're talking about. So if you're talking about pain, it might be a different story. But in Alzheimer's disease, the actual, because a patient received the placebo, they get better, that effect is not enormous.
All right. I have a ton of questions on regulatory, but instead of asking you them now, I'm going to advise the audience that later on in this webinar series, one of the webinars is going to focus on regulatory. So there's so many of these questions over here, and we don't have that much time. There's so many other questions that I would like to just push on to some of the other questions over here. And I do want to just remind the audience, we are going to spend an entire webinar on the regulatory stuff. I know something that I also want to ask Charles as well. But let's move on to data privacy, because there's several questions over here, and I'm just gonna, they're all asking pretty much the same thing here. It's basically, what are the implications for patient consent? How much information about the digital twin and the projected outcome is disclosed to the actual patient?
Oh, that's an interesting question. Okay, so if we go back to the beginning of our training data sets, and actually you contrast this with EMR data, right? We are starting, another example of an advantage, we're starting with data from patients who consented to take part in research studies. All of these data are consented for this purpose of research on this disease. It's not data that we're just taking that people don't even really realize in some cases is being used, right? So these are data consented for research. So that's like our foundation. Then we are actually only using de-identified information. We don't need the patient's name or address or anything like so we're creating de-identified things. And then of course we have to take data security and all that kind of stuff very seriously in trials as well. The last question in terms of disclosing the digital twins to a patient is, whoever asked that question is a very interesting question. I had never thought of it before. I just have to admit, I had never thought of that before. So that's a very interesting question. So yeah, it's something that we will have to think about. I think one of the things that we thought about as a potential future application, which is outside of clinical trials, right, is if you had this ability to do digital twins and to get simulated progression of people, maybe that would be useful to them or their caregiver or their physician for different types of things. And maybe that's a potential future application. Again, for Alzheimer's disease, it's tough today because it's not clear what you would do with that information because we don't have treatments available that are disease modifying. But if you start talking about other indications like our work in multiple sclerosis, where you do have disease modifying therapies, that question does start to be more interesting. It is interesting in Alzheimer's disease, but there starts to be something you could do about it. And so, yeah, that's a really interesting question. Thank you, whoever asked that.
Oh, that's a great question. There's one comment I would like to read real quick to you, Charles. Thank you for such a clear and easy to follow presentation. As a patent attorney, I don't work directly in the field, but I found this really fascinating. There's a couple other questions here that we just received on what are the dates of the following webinars. We've got one up on October the 21st. We have one coming up on December the 2nd. But anyone who's registered for this webinar is going to get a follow-up email from Endpoints News, from Unlearn, with the direct registration link to all that, so you don't need to worry. You can also visit webinars.endpts.com and view Endpoints News' entire webinar lineup over there. Let's move on to a couple more. We only have just a couple short minutes here left, but I'm glad we have you for another 2 webinars, Charles. Have you ever rescued a trial? Have you ever rescued a trial after the fact by increasing the numbers after the fact?
Yeah, that's a very interesting question. We don't have actually any trials that have completely finished right now. We are using this approach in a phase 3 clinical trial in Alzheimer's disease now. So but it hasn't finished yet. So it's ongoing. In terms of rescuing trials, one of the things that we've thought a lot about is not necessarily rescuing trials completely after they're done, but looking at trials right now that are impacted by low enrollment or poor follow-up because of the pandemic, right? We're all stuck inside, especially people in elderly populations who maybe cannot make it to the sites because they're at high risk. Alzheimer's clinical trials have been impacted a lot by that, as have other clinical trials. So you have these trials that have extremely low enrollment that are going to be underpowered because there are lots of dropouts and poor follow-up and low enrollments, and adding these patients into those trials, adding intelligent control arm can help to recover and make it much more likely that those trials could find an effective if that drug is actually effective. So that is something, and we're discussing those kinds of things with a number of different potential partners now.
Okay, which actually leads me to one of my last questions over here for you. We'll get to a couple more here, but quickly, can you just talk about some of the work you've done with research and with partners? You've talked about that one phase 3 Alzheimer's trial.
Yeah.
What else can you tell us?
Sure. Well, the biggest thing that I would tell people to do is to tune in to the Clinical Trials on Alzheimer's Disease Conference, which is coming up in the first week of November. We're giving 2 presentations there, which would be detailed retrospective analyses in collaboration with an academic Alzheimer's trialist. So we're going to be presenting a bunch of research on this soon. In terms of fundamentals of like the machine learning and its application to different things, we have a whole bunch of papers. Again, if you go to our website, the work in Alzheimer's disease, we published a paper in Nature Scientific Reports. That came out last September. And of course, it was written like 2.5 years ago because of how long it takes peer review to work. So that's a very old version of the model, working on a much, much smaller data set than what we're working with today. But it does describe the same methods. And then we also have a paper describing a similar approach in multiple sclerosis. You can find a preprint of that paper that's posted on our website and on bioRxiv, but that's currently undergoing peer review. So we hope to hear back about that soon.
Great, great. Okay, I'm going to try to get to 2 questions here. So second to last question here. We'll try to keep it quick. How can digital twins assist in trials impacted by COVID-19?
Oh, well, I just said that kind of, but yeah. So low enrollment of your trial, poor follow-up, all of those things that we can add an intelligent control arm. Again, all we need is the baseline data, and that can basically recover maybe all or maybe even more actually of the statistical power for your trial. So depending on how impacted it is, you could actually end up getting more power than you had initially planned.
Okay, well, let me ask you another quick fire question. If we do not have data ahead of a trial, could we design a trial with less real subjects, say half the size of the PBO group, roll that N into enrolling more actual treated subjects, and then use the digital trial methods to use half the PBOs to double the PBO group? Do you follow?
So if the first premise of it though was that there's no data to train the model.
If we do not have data ahead of the trial,
Yeah.
Could we design a trial with less real subjects?
Sure.
Roll that N into enrolling more actual treated subjects.
Yeah. So if we have a model already trained for an indication like Alzheimer's disease, then you can design a trial that has fewer subjects in it. And it can have a much higher randomization ratio where the majority of the patients in the trial get to receive the active treatment. However, if there is no data, if we don't have a model and there is no data to train it, the data do have to come from someplace, right? So the model has to have learned from something. And so there has to have been some historical data. And again, if you're looking at, if there is no trial data available, then maybe you could just use registry or natural history data, right? So if you're looking at like a rare disease, you might be able to just leverage natural history data, but there does need to be some data available that the model can learn from.
Okay. All right. We got just 1 minute left, so I'm going to have to ask you to keep this answered about 45 seconds, Charles.
All right.
What's your vision for digital twins in the next year or next 5 years?
Yeah, well, the next year, we're rolling this out on Alzheimer's disease. And we believe this is just a fundamentally better way to run clinical trials, right? You get the benefits of an RCT with the benefits of a single arm trial, and you don't have the downside. So we think that this should be rolled out into essentially every clinical trial in that field. And so once we really prove this out in Alzheimer's disease, one of the things we want to do is build trust with trialists, build trust with regulators, build trust with the consumers. And once we've done that, we'll start to expand into new indications.
Right. All right. Charles Fisher, thank you so much for such an engaging and awesome presentation. We have 2 more of these coming up: AI, digital twins, and the future of clinical research. A couple more coming after this. I'm really excited for that. Charles, we'll see you again. And for the rest of the audience, we are going to follow up. This presentation is going to be on demand, and we hope to see you again. Charles, thank you very much. I'm Arslan with Endpoints News. See you later.
Thank you very much.